Miss Litton, and this is AP Biology Review of Chapter 37, Neurons in the Nervous System. Say hey.
Hey.
All right, very good. All right, the only thing I really wanted you to see in the beginning, I wouldn't spend a ton of time here in the beginning, okay? Just the thing I wanted you to see is the what of a system. Evolution of a nervous system. So we could see here with sponges at the cellular level of organization, they don't even have tissues, right? They haven't even graduated to that level yet. They work together. We saw nerve nets. We saw in planaria about 2 nerve cords. Keep in mind, the more centralized it is, probably the more what it is. Evolved. It is, okay? We worked our way up to groups of cell bodies called ganglion. We saw ganglion, our dorsal ganglion outside of our spinal cord. What are in our dorsal ganglion outside of our spinal cord? Sensory neuron cell bodies? Yes. All right. We looked at vertebrate nervous system. We looked at the forebrain, the midbrain, and the hindbrain. If you look at things like that are part of the hindbrain, the pons, the medulla oblongata, cerebellum, what are we thinking about? Those types of things. What do they do for us? What type? Are you saying autonomic? I agree. Okay. Even our midbrain doing that as well. When we think about the forebrain, like this, minus the hypothalamus, when we look at the cerebrum, we're thinking of memory, learning, thought, understanding, right? And we could see that when we look at the evolution of the nervous system up through mammals, that you have more focus and larger cerebrum. We then broke down our entire nervous system to our central and our peripheral nervous system. So you should be at this time able to go, okay, central nervous system consists of brain and spinal. When I think spinal cord, I think, what are they in charge of? Reflexes, right? And also bringing information to and from, right, to my brain. Because how many pairs of nerves do I have off my spinal cord? 31 pairs, and they are all mixed nerves, carrying both sensory and motor information. Yes? Okay, so they're going to bring that information here to my brain, but reflexes, I don't even have to process reflexes. I can automatically respond. My central nervous system, I have all those things like balance and hypothalamus and breathing, all part of my autonomic, and then my cerebrum, memory, learning, thought. We can break down, we already talked about cranial and spinal, sensory and motor, we understand that. What is missing? There's a neuron that's missing here. And where is that? In the central nervous system, right? That's where your inner neurons are? Is that what you were saying to me? Of course, all right. Then somatic and autonomic. Somatic are things that I what? I control them all, but I consciously control. I'm making a choice. I'm choosing, right? I'm choosing for my somatic nervous system unless it's a, yeah, then my body will take over for that, right? My autonomic nervous system, I've got 2 categories in my autonomic, right? And it's, what are the 2 categories?
Sympathetic and
parasympathetic, okay? So, and you know, sympathetic, what would be a phrase you would use for sympathetic? One word, you could use that one if you want to. Fight or flight, anytime stress, right? Your tigers today are things like tests, you know, sporting events, right, that you have to compete in, somebody yelling at you. Those are your tigers of today that give you your stress, your fight or flight. And then what would be the one for parasympathetic? Yeah, and then there's some like certain subtleties out of each of those that we can dissect again a little bit. All right, we talked about staying in our large mammalian brain, not just in our, trying to and use logic and thought, and that's good. Okay, neuron. When you think about a neuron, there's a couple things I want you to think about. The types of neurons, which we just went over, sensory, motor, and inner neurons. Then I want you to look at structures of neurons and be able to recognize all the structures. So what's this part that receives? What's that called? Dendrite. And it could be attached to the, if the cell body and the nucleus are involved in processing. Who's the exception to that rule? Sensory neurons, exactly. That's what you're saying. I know that's what you're saying. Sensory neurons would be the exception to that. Am I actually recording? Did I hit start? Okay, I did. So sensory neurons would be the exception because you either at the dendrites reach the threshold or you don't reach the threshold, and that neuron either fires or doesn't fire. Then we have the cell body, the nucleus, and this is called what? What's the long part of it called? Axons. And bundles of axons are called what? Starts with an N. Nerves. Bundles of axons are nerves. Some nerves are sensory nerves, meaning they only have neurons going towards the central nervous system. Some are dedicated motor, only going away, and some are mixed. Okay. Also, you could have this. What would you call this in general? Myelin sheath. Who creates the myelin sheath in the peripheral nervous system? Schwann cells. Good. How about in the central nervous system? Oligodendrocytes. Say that word. Oligodendrocytes. Okay, and then the ends of axons, what would you find at the ends of those axons? Synaptic vesicles. These are like little baggies. Synaptic vesicles. What's inside the synaptic vesicles? Neurotransmitters. Those are chemicals. Those chemicals that are released could either be or exactly. Okay, excitatory or inhibitory. So then we looked at also support cells. Here's another one of support cells. And astrocytes are kind of feeding, oligodendrocytes, we already talked about myelin sheath. Then we have other ones that are going to phagocytize and keep it clean. There's actually more support cells in your central nervous system than there are cells that you're thinking and processing. Okay, so those are the support cells. There was another one I wanted to show you a better picture of here. Okay, ependymal cells, those are they make the fluid, the cerebral spinal fluid, right, that runs around the central canal and bathes your central nervous system. We already talked about oligodendrocytes. Okay, I'm good on that. Here. Now, looking at this picture, we looked at this at the beginning, but then we talked about something here at the bottom, and this should make sense to you what's here on the bottom, right? So sensory neurons going towards the central nervous system, motor neurons are going away. There's only two targets. Who are two targets? Who are your effectors? Glands and muscles. So this one's going to your skeletal muscle. So what system is that a part of when we divide our peripheral nervous system? Somatic, right? Skeletal muscle, I'm choosing to move, right? That's my somatic nervous system. Smooth muscle, cardiac muscle, glands, that's all what? Autonomic. And what are the two divisions of the autonomic nervous system? That's right. And in your autonomic nervous system, you have two motor neurons. The first one should be myelinated, the second one should be unmyelinated, but they oversimplified here, okay? In the sympathetic nervous system, the first motor neuron is short. And the way I think of that is they're in a busy, it's fight or flight, it's stress, so quickly do your synapse and move on. That's how I remember it. Parasympathetic, rest and digest, it's like, oh yeah, I better synapse here. Now can you think of why that would be an advantage to have a difference where they synapse if there's two motor neurons? Why would that be an evolutionary advantage to have two different locations where they synapse? Less confusion, less chance of crosstalk. You're not changing, whether your heart beats faster or slower, you want to be sure about that, right? Okay, so if they're synapsing, if there's two motor neurons and they're synapsing at the exact same location, you could have some issues there. So that's adaptive, right? That they're synapsing in two different locations. All right. Okay, we'll revisit that when we go to the ones here at the end. That is boring. Okay, let's go here. All right. So, and again, I recorded two separate videos on this that you could see on my YouTube channel. One just about the synapse and one about the action potential. So let's just think, when a neuron is at rest, the inside is charged to what? Negative 70 millivolts. Perfect. What has created that? Number one, Sodium-potassium pump. 3 sodiums out, 2 potassiums in. Number 2, negatively charged ions and proteins inside. Number 3, leaky potassium gates. Okay? So at the end, we're more positive outside and more negative inside. Okay? A neurotransmitter, let's assume it's creates an EPSP, which stands for excitatory postsynaptic potential. Okay, this neurotransmitter is going to generate an EPSP. If it's going to do that, then you have what? Receptors for the neurotransmitter. And these are ligand gated, right? Some chemical is opening in, not a factor of charge. So let me jump to the very un momento. Here. Okay, so at the end, here we have some chemical, ligand gated, binds to it, opens up. Who wants to come in through facilitated diffusion? Sodium. Sodium goes, sure, open the gate for me, I'm in. It goes in, it makes the inside less, and makes it more, which then can trip the voltage gated, right? If you're closest to the axonal hillock, the closer you are to that, do you see you have more of a vote because you're letting more positives in right where you're going to fire it. Okay? If that happens, then sodium comes in, brrr, right behind it, who's fixing it? Quick fix. Potassium going out. But either way, you're getting a chemical message is triggering an electrical signal along the length of that neuron, and then it's getting it to release chemicals at the end. So you're getting from point A to point B, and that's an electrical message. So what happens there at the end? Sodium comes in, yes, potassium's doing a quick fix, and right behind that, going all along, what's the long-term fix? Sodium-potassium pump. But down there at the end of the axon, instead of sodium coming in, now what's coming in? Calcium's coming in. Calcium has the ability to bind to the synaptic vesicles and cause the synaptic vesicles to migrate to the end of the presynaptic neuron and release it into the synapse or the synaptic cleft. Okay, so the cleft is specifically the space between the two neurons. The synapse is the whole thing. So let's release it into the synaptic cleft, that little space right there. It'll cross the synaptic cleft, and if it is an IPSP, it's going to keep the doors closed. It's not going to let sodium in. It could make it more on the outside. What? More positive on the outside. Or if it has alternative doors to open, it can make it more what? More negative on the inside. That would inhibit that neuron from firing. Does that make sense to you? Okay, turn towards your bio buddy that you're sitting next to right now. Say questions or clarification. Anything confusing on that? Ask them. Okay, come back to me. Any unresolved issues you would like to discuss? I do not want you to leave here today with any confusion in your brain. Good luck. No, anything in there that you need me to work on. Get in your brain. Nothing? Okay. All right, let's keep going. All right. So when we talk about synaptic integration, and by the way, if you want a good picture to capture the synapse, that tells it to you in sequence. If you had an essay to write, let's say on a synapse, might be some things you're missing.
Thank you.
You're welcome. You had this in class too, remember?
Yeah, just didn't take a picture of that. Just in case I didn't take it back then.
Okay. All right, we ready? All right, integration, synaptic integration. Synaptic integration, what could that mean? I'm at the synapse, I'm integrating. So that must mean I'm adding up all my boats, the excitatory and the inhibitory. I'm adding them up, and it still doesn't mean I'm go for launch unless I cross the threshold. Then I'm ready to go. Okay, and did I pass that one? I did. Speaking of that, that's a great diagram too to know and understand. Okay, this is our resting potential, minus 65 millivolts, created by the sodium potassium pump. Negatively charged ions and proteins.
And
Leaky potassium gates. What must be happening right now? What's happening right here? Sodium's coming in. Come in, sodium, and it's making it less negative inside. And as soon as we cross this, then for sure that neuron's going to fire. Okay? They don't fire with any greater strength. They fire at the same speed every single time. There's consistency in that. It's just fire or not fire. That's it. Your light is on or light is off. There's not a dimmer light. Okay? Neurons fire or they don't fire. So this is sodium rushing in. That is depolarization. Repolarization is caused by potassium rushing out. What's happening here? It's oh so negative. What are you saying? Overshoot, refractory period. And this is really good because this keeps the action potential traveling in just like and a valves in a vein. Exactly. Keeping it going in one direction and then returning to that resting potential. You're so smart. So smart.
On the way down, what's happening? Like when it's
on the way down, what's happening? What's bringing it down? I skipped over that. I'm so sorry. Of course, I can't find it now. There you are. What's happening here? What's bringing it down? You're bringing me down. Potassium's going out, right? Because here, let's think about this. We have plus charges inside, right? We do have plus charges, but we have more plus charges outside. So the inside is negative, yes? But now we're getting double positives in there because we have sodium and potassium in there now. And then potassium says, you're in here, sodium? Then I'm leaving. I'm the quick fix. So potassium takes its place outside, and now we're returning to being more negative inside and more positive outside. You okay?
Yeah.
All right. Okay. No. Let's talk about the central nervous system. Brain, spinal cord. It is protected by bones. Okay, bones. Not, right? Bones, tier 2, right? What else is it protected by? Meninges? Say it, meninges. What's the first one called? Pia. Arachnoid. Dura. It's very durable. Okay? So it goes bone, meninges, and fluid. So there's the fluid. I have a picture. Here it is. Pia, arachnoid, dura on the outermost. Really 2 layers of dura. Okay? And then fluid. Good. Now, this is a great picture. This is a great picture. This person is looking at me over here. Right? Where's my butterfly? Okay? What's going in down at the base of the butterfly, farthest away from me? The back of the spinal cord. That guy's spinal cord. What? Sensory neurons. Sensory neurons go in the back. Okay? They sneak in the back. Sensory neurons sneak in the back. Motor neurons out the front. Motor neurons out the front. Okay? Sensory neuron. Now, there's a sensory neuron in here going like this, right? Here in the ganglia is its cell body, and it's headed in this direction, right? Headed this way, headed this way. The motor neuron is coming out the front, headed this direction. Its cell body is right here. What could be in between those two? Inner neuron. Okay? The inner neuron's right here in between those two. Gray matter is where cell bodies are. White matter is where you have myelinated, neurons, and that's why it appears white. Okay, but this nerve right here, it doesn't just have these 2 neurons in there. There's multiple neurons in there. A nerve is multiple long axons. But when you grab, if you could grab somebody's nerves on either side of their spine, they would all be mixed nerves. How many pairs? And if they said, I'm going to hit you in the back with a hammer, are you going to say aim high or aim low?
low.
Aim low. The higher the injury, the less control you keep, right? I probably showed this picture in some classes, yeah? Did I show you this picture? Okay, here's a picture for you. Do you understand that picture? That's why I say aim. Do you see why I say aim low? Okay. When you look at this person's face down like this, and we've chopped them in half, and you're looking down their spinal cord. Okay, so what's happening here? An in or an out? In with sensory neurons, and down here, out with motor neurons. Good job. Okay, you can see here, ascending track would be sensory neurons going up to the brain, and descending would be motor neurons going away from the brain. All right, what else? Okay, the parts that are in green that I've highlighted, I think those are good ones to know, just general brain things to know. So we can review those. And we probably don't even need slides for it. Okay, let's start with the cerebrum. What does the cerebrum do for us? Memory, learning, thought, right? Accepting sensory information, processing, sending out motor information. Yes?
Yes.
Okay. What's the corpus callosum? It's already here. What's the corpus callosum?
Nerve fibers connecting.
Connection. Good. All right. Diencephalon, there's two of them, thalamus and hypothalamus. Thalamus, okay? Of your senses here, what do you think is more critical, smell or taste? Smell. Remember you did a video on that, right? You can detect more smells than you can detect taste, right? Okay? And so smell is that ancient primal chemoreceptor that we have the most variety in. Do you remember what those cells are called here in your nose? Olfactory. Yeah. You know, I remember that? Like old factory, it smells. It smells like an old factory. Okay? Now, you're like, no, I just remember it. Okay? So, it's primarily here, what it looks like the fried egg there, that thalamus, where you connect smells and senses with memories and potentially emotions. That's, if you think about it on the evolutionary scale, that's a food I can eat. I know that makes me sick. That smells like a mate. Okay? So all those kinds of things help you in your survival skills. What's the significance of the hypothalamus?
Homeostasis.
Who does it work with? Endocrine gland. Who's the master gland? Pituitary gland. It's right here. Okay? And so that's where the two talk. We're going to learn about that tomorrow. Cerebellum, what's this about? Balance, right? And muscle tone, coordination of your movements. Midbrain pons, these are all about, relaying information back and forth. Medulla oblongata, super important. What does that do? Breathing rate, heart rate. Yeah, so you know them. Notice here, all those folds on your cerebrum, or cerebral cortex, those are going to increase what?
Surface area.
Look where the gray matter is in your brain. On the edges. You see that?
Yeah.
Different than the spinal cord. That's where those cell bodies are going to be. That's where you're making those connections. Next. You know this. This. I talked to you about reticular formation. Right here. I can share this out with you. We can do it right here. It makes no difference to me. Can we share it out to you? B. Your favorite color. My favorite color is Becky. My favorite color is Becky. Favorite color, Becky.
Harry Potter had a color.
Green, or is it green? It's green. It's green. Okay. Harder. Black, the color of my soul. Okay. This one is name that part, and then you click on the box, and it'll tell you if you're right. Name that part. See if you're right. I'm going to pause you just for a minute. Or here, I'll do it for you.
Oh, you didn't think about it.
What's this one? What's the yellow one? Yeah, it's your cerebrum. What's the red one in the top right? Thalamus. What's this one over on the right? I'm going clockwise. hypothalamus. What's this right here? Pituitary gland, spinal cord, medulla oblongata. Hey, medulla oblongata is in the wrong place. Medulla oblongata should be down here. Oh, I'm so glad we picked that. And cerebellum. Yeah, medulla oblongata is in the wrong place. Okay, there's another game here, but I'm going to keep reviewing. If you want to do the game at the end, I'll do the game with you. There's another one on here. Okay, a nerve is a bundle of long axons together. We know peripheral nervous system. How many pairs of nerves come off our brain? 12. 12 pairs.
12 pairs of
Sensory, motor, and mixed. Yeah. Here, this picture should be no problem. You should understand exactly which way this person's facing. Where are they facing? Face up or face down? You give you a big hint here. See this? Feel your back. So how are they facing? Face down. Okay, look at the butterfly. Sensory comes in the back, motor out the...
Wait, why are they facing?
Your bones or protuberances? Yeah. Got that? Okay. Next, this picture. You want this picture? You understand that? Okay. Sensory information. Here's the dorsal root ganglia right here that have the cell bodies in them. Yeah. Okay. And we're synapsing here with inner neurons and synapsing with motor neurons that are coming. Whoa, hello there. That are coming out the front, right? Motor neurons are going out the front. All right. Next, reflex arc. Okay, so you have a stimulus, sensory neuron, synapses simultaneously with an inner neuron and a motor neuron, which exits out the front, so you can pull away simultaneously by being aware of what you're pulling away from, right? And that saves time, so that you respond quicker to whatever is coming at you. We talked about this. Yep. So me kicking a soccer ball, what am I triggering there? Kicking. Who's my somatic? Okay. Me running while I'm kicking that soccer ball is going to be what? Not somatic. Oh, sorry. You're right. Running is my choice.
Yeah.
What do you mean? Okay. My heart's beating faster because autonomic what? Sympathetic. Does that make sense? Yes. All right. And then here we can see sympathetic nerves come out of the middle of our spinal cord. Parasympathetic out either end. I think like they're in the middle of it. Adrenal medulla, fight or flight. That's how I remember it. But then I want to talk about those two motor neurons, right? The first one is short, second one is long. For parasympathetic, first one's long, second one is short. What's the exception? Where don't I have a second motor neuron? Do you remember where? To my adrenal medulla. Okay? There is no second motor neuron, and that's because my adrenal medulla, my adrenal gland, just might be an enlarged second motor neuron. Because what comes out of your adrenal gland, adrenaline and noradrenaline, is the neurotransmitter of your sympathetic nervous system. Right? We're all a bag of chemicals. So the chemical that our hormone, our endocrine system uses, is the exact same for fight or flight, is the exact same thing as our nervous system uses in our sympathetic nervous system. Okay? All right. I think I have some questions.
Yeah, we had a lot of questions.
We have a lot of questions. Let's start with these questions. I will pause you. Take a look at it. Because all the spinal nerves are mixed. All right. Number two. Oh, one person. Man. Okay, there's some more here. Did I let you play this? Did we do rock band?
Rock band.
Rock band. Some volunteers to do it on the board so it'll be recorded. Come on. Do it with a friend. Bring your bio buddy. Do it on the board so that it'll be recorded.
So, the hypothalamus.
I'm not tall enough. This is sad.
Hypothalamus is for homeostasis. Oh, okay. I only do one?
No, you do the gray and you match with the red. To help her match though.
Spinal cord.
Do it wrong.
Oh, come on. I'm also going to do this
Balance is cerebellum.
Cerebellum?
Yeah, it's the lower one.
Cerebrum's over. No.
No, it's at the top.
This goes to the top? This is part of the thing then. Okay, limbic system.
Let's go with the right.
Wait. Reticular center, that's easier. So that's probably memory and learning. Oh, okay, now I know what it is.
Told you.
Gee, I wonder what this last one is.
Okay, here you go. Here's another one. Pause it just for a second. Okay, you guys, hurry up, make a guess. All right, here we go. Let's check it out. Good job on number 1. More sodium ions outside, fewer potassium in. Good job. Good job. We did this one in class for some of you. I remember this. So, we don't have the release of neurotransmitters at the postsynaptic membrane. We receive neurotransmitters at the postsynaptic membrane, and we don't release sodium ever, right? Sodium does facilitate a diffusion in. It's not released. Like, release the hounds. We don't release any sodium. Yes?
For B, even if it did say the presynaptic membrane, the actual release wouldn't actually do anything until it is received. So
Right, but you could say the release of neurotransmitters from the presynaptic membrane. That's how I would say it. Okay, I think there might be another one in here if you want to review it. I think.
I remember doing a lot of them in class.
That might be it, I guess. Unless you see one, I don't. There's a monster quiz in here. There's also kind of a slow game, I think. Maybe I took it out of here.
We did the monster quiz.
We did the monster quiz. Okay, we're good. All right, make good choices. I'll see you mañana. Have a piece of toast if you're tired.